The overall goal of this procedure is to make use of clotted bone marrow samples by digesting the clots with urokinase. This is accomplished by first filtering the bone marrow sample over a cell strainer. Next, the bone marrow clot is cut into small pieces. Then the clot is transferred to a falcon tube and digested using urokinase. Finally, the digested clot material is seeded for expansion of mesenchymal stem cells, or MSCs. Ultimately, the plates are compared to a control plate using Giemsa stain to assess the efficacy of the digest.
The implication of this technique extends toward autologous stem cell therapy or diagnosis, because grafts need expansion and differentiation of a large number of stem cells.
We first had the idea for this method when we realized that a lot of bone marrow samples from dogs were clotted when they arrived in our lab. We simply did not want to trash such precious samples.
To reconstitute urokinase, inject 10 ml of sterile PBS into the septum inlet flask and vortex to dissolve to a concentration of 50,000 units per milliliter. Aliquot 500 microliters of 25,000 units each into sterile tubes and store at -20 degrees Celsius for up to 6 months. To carry out a digestion reaction, thaw 1 aliquot of urokinase per clotted bone marrow aspirate of up to 25 milliliters. Preheat a water bath or shaking incubator to 37 degrees Celsius. Under a laminar flow biosafety cabinet and while wearing gloves, place a 100 micrometer